So yeah, next speaker is another former student, Dr. Jahnavi Joshi. She was a student in Dr. Praveen Karan's lab, and Jahnavi represents the first generation of students that Praveen and I had after we joined CES as faculty nearly 20 years ago now. Yeah, and I think there was till Praveen and I joined, there were five or six faculty when before in a fairly small student community, and that sort of like exploded. I think this was the largest batch we'd ever had. There were 11 students in the batch, and yeah, I mean that kind of changed the face of the department, I think, quite dramatically. Plus, as Manjari said, she said me, but it was me and Praveen walking around, and they dragged us quite a lot. So yeah, Jahnavi, please.
Well, thanks, Karthik, and thanks everyone thanks Karthik, and thanks everyone at CS for having me here. Still not, yeah. And as Karthik mentioned, I'm a student from 2006 batch, and still not. Okay, now. Is it okay now? Yes. And much of the research which I do and as a group we do is very much shaped by my time spent here at CES in the lab. Discussion on macroecology and microevolution was emerging for me, and maybe if I'm very bold, I'd say was the kind of beginning of thinking about biogeography, linking ecology and evolution together while understanding diversity patterns was something the research was initiated by both Praveen and Karthik's lab. So one of the main research themes since I started my lab at CCMB for the last 4.5 years, we have been thinking about how do you link a continuum of microevolutionary processes where we are thinking about linking processes which operate, the drivers which operate within a species across populations. Then we think about how a speciation process which forms a new species operates, what are the drivers of that, and then ultimately the speciation process, diversification, leads to the patterns which we see today as the diversity, and how is this diversity generated over large spatial and temporal scales. So today what I thought I would do is talk about the later part of it, where we are thinking about what are the evolutionary drivers of tropical diversity. And as my first image showed, I'll think about from peninsular Indian perspective, very much work focused on the Western Ghats. I'll introduce the kind of conceptual framework and peninsular India very briefly, and then show you a couple of case studies where we are trying to understand what are the diversity patterns within the Western Ghats and what could be the potential evolutionary drivers of those. One of the most striking patterns for all of us is when we look at the diversity maps is that tropics are rich. And this has fascinated many researchers to ask very fundamental questions. How is this diversity generated and maintained over long evolutionary timescales? Some of the explanations are with the age of the tropical biome, the stability of tropical biomes. They have argued that tropical biomes are much more stable, so species could accumulate. They may accumulate at a slower pace, but to accumulate over a longer period of time, you have a high diversity, which is depicted in this phylogenetic tree where what you have is the long branch length, ancient species getting accumulated slowly. On the contrary, you could also have a credit or a speciation, very high rate of speciation in these tropical biomes, and that's why you see high diversity. Why so? When you overlook the mountains, you see many mountain ranges in the tropical forest, and they offer a large variation in climate and topography over a small spatial scale, and this gives you a micro-refugia in the times of climate instability, as well as an opportunity for ecological speciation. We also understand now, if we overlay the biodiversity hotspots, where are they? They are in these mountainous regions in the tropical forest. We understand what have been the tectonic history of this landscape which we have shaped. Within the tropical belt, when we think about it's a climate which is similar across its tropical area, you have a disparity in diversity. It's not evenly distributed, and it's not across taxa as well. And what could be the potential drivers of this? People have argued the strong regional biogeographic history of that region, the speciation and extinction rates in individual organisms are driving these factors. And in this context, when we think about South and Southeast Asia, our understanding is kind of limited. We are still understanding what are the biodiversity, diversity gradients or factors there are. And then we are going to think about what are the potential processes which may be driving these. I know this audience doesn't need an introduction to the Peninsula of India, but it's one of the oldest landmass in Indian subcontinent and Southeast Asia or Oriental realm, known as the oldest region for diversification. We have one of the global biodiversity hotspot, the Western Ghats, the escarpment which runs along the west coast of India. You have Eastern Ghats, which is much more broken chain of mountains on the east coast of India, and you have very diverse habitat types. What has been the biogeographic history or tectonic history of the Indian plate? You know, it was a part of a Gondwana landmass, sequentially it broke away from other continents, moved a largest distance as an island for a long time. During that period, it experienced a Deccan volcanic activity, which led to mass extinctions, drastic climate changes, collided with Asia, Himalayas came about. You also had a monsoon setting up, it's intensified aridification, and lastly, last glacial maximum. And undoubtedly, this geoclimatic history has shaped the diversity which we see around. And now we are going to ask the question, how do we then study how this geoclimatic history, the current topographic heterogeneity has shaped the diversity patterns which we see. I want to use a phylogenetic framework which uses molecular phylogeny, which allows us to think about how this potential processes may have given rise to these patterns which we see today. Here is a tree which has both older lineages which are depicted in these longer branches, shorter branches, younger lineages. And if I want to associate what sort of phylogenetic patterns you see to a process is the exercise we are interested in. To do that, I want to use two kinds of approaches. A special phylogenetic diversity measures, which will allow me to think about this evolutionary history of phylogenetic tree and measure the phylogenetic diversity, and then actually explicitly measuring speciation and extinction rates and their contribution to this diversity patterns. Very, very briefly, what is phylogenetic diversity is nothing but a summation of branch lengths of the species which occur there. For example, if this is the phylogenetic tree, which is a time calibrated, what it means, how these species are related to each other, who have their common ancestry. If this area has a 4 species, this is the phylogenetic diversity, which is a summation of branch length. Area B has 4 species which come from very distinct phylogenetic tree, that becomes a phylogenetic diversity. If we scale this diversity, where do they occur? How many grids do they occupy? That becomes a measure of endemicism. Is it a widely distributed species or is it range restricted species? So you can have now thinking about, you can be a long branch species, ancient species, range restricted. You could be a younger species but widely distributed or both. People have been studying this across the bio. These are two phylogenetic diversity maps. This is for plants, this is for snakes, birds and lizards. Now, one group which I continue to work on is arthropods and which are missing from the global analysis. So we decided to tackle this group because we have been working about it. We know how many species are there in the Western Ghats, where do they occur, and their phylogenetic relationships. On the contrary, is a very diverse group. We have distribution data. We understand their diversity, but we didn't have a good handle on their endemism patterns. The questions we asked: how is the diversity distributed in the Western Ghats, and what are its potential drivers? Are the Western Ghats are really museums of credit? What sort of species do we have? We have old ancient species, or we have young range restricted species, or a combination of both. So I want to present two case studies, one using soil arthropods and one using plants. Solomated centipedes is a work which I began when I was in Pravin's lab as a graduate student. We understood how many species are there in the Western Ghats, how old are they, where are they, what sort of biogeographic patterns do they show, how have they diversified within this landscape. We know there are Gondwana species, we know they are diversified here, we know that they are endemic, we understand where they are distributed. But when I say this, we still have a very limited understanding of this group. Bharti, when I set up the lab, one of the first person who joined my lab was Bharti, who is an alumni of CPS 2009 batch. So I'm going to talk about her work now. Thinking about species ranges is not a trivial thing. We had distribution records, but that didn't capture the species range. So Bharti used her expertise of species distribution models. We set up the protocol, we built these models, opened it to get a diversity gradient. We had a phylogenetic tree where our sampling was close to 100%. It was a dated tree, so we had taxonomic, like we could actually measure both these measures using time calibrated tree. What did we find? Like many other taxa, we had a latitudinal diversity gradient. Lower latitudes had more diversity as one moves towards north, or higher latitudes diversity decreased. We also had older species in south, younger species in northern western Ghats. When we looked at their ranges, south was a signature of a gradient where you had range restricted, high diversity. Interestingly, species poor areas of northern western Ghats popped up because they had very few species, younger species but still range restricted species unique to those landscapes. Moving on from this work, this was 20 species, very species poor data set. These are predatory soil arthropods, so they are not very speciose groups to think about it. Then we moved on to woody plants, and this is the work led by a graduate student in the lab, Abhishek, in collaboration with Navendu, who is also a CS alumni from 2008 batch. What we did here is looked at the data which Navendu had collected during his PhD across the Western Ghats, complemented that with other occurrence data, used the similar approach which was developed for scientific work. We used the global tree which was available for plants, looked at what are the diversity patterns. We see a similar trend. Diversity decreases as one moves towards north. Interestingly, plants also have older lineages which are throughout the Western Ghats. So this is in accordance with the museum hypothesis, where you have older lineages throughout the Western Ghats. South has lots of younger lineages, range restricted species, along with these older lineages. So Western Ghats acts as a both museum and terrain. It's not either or. You have old lineages which are widely distributed. You have younger lineages which are range restricted. However, these still and speculated on the processes which are driving this. Now is the time to really think about what are the speciation and extinction rates which may be contributing to these diversity patterns we have seen that. Pragyadeep joined as a lab as a graduate student who also happened to be a CS alumni from 2019 batch. Pragyadeep did this work, and this really built on a lot of work which has happened in CS across last 20 years, where we had time calibrated trees, not only for one or two groups, but multiple groups across taxa. So we built on this. This is the data set. We looked at 33 time calibrated trees, which span across angiosperms, herpetofauna, invertebrates. The two questions we asked: what sort of patterns do we see? Do we see elevated patterns in general in plants? Because people have argued that they have large range sizes, they show polyploidy, their dispersal modes are different. So you may have higher diversification rates or speciation rates as compared to animals. Is that the one reason we see higher diversity across patterns? We also have species with very distinct biogeographic histories in India. You either have an Asian origin or a Gondwanan origin. And interestingly, what we see here, the Gondwanan species tend to retain their ancestral niches, which are wet forest, versus younger Asian lineages have diversified throughout Peninsular India. If that's the case, you will have a higher rates of speciation for younger groups and a low speciation rate for ancient animals. Does it matter where you are, wet forest or a dry forest? Do you differ in your diversification rates? Then we asked, how is the diversity accumulated? Remember, I started off saying one of the main hypotheses for high tropical diversity is the old biome and it's stable. So, if you have an old and a stable biome, what to expect is diversity to accumulate slowly and it will show gradual accumulation of a diversity. Is that what we see in the Western Ghats? And that's what has led to the diversity we see here. Or Or is it actually responding to the changes this landscape has faced throughout its geological timescale? So you could have something called saturation, because now all niches are filled, you don't diversify anymore unless some drastic climate change or other thing happens and the niches open up. And you see a shift. Do you see a constant oscillation in the speciation and extinction rate, waxing and waning pattern? Or suddenly new niches have been now opened up. And it could be any geological event which is doing that, or any other key innovation which happens within a group. And you will have something called adaptive radiation of high diversity in a very short period of time. And then, if these are all the things which are happening, how does it relate to the species richness pattern which we started thinking about? To do this, we used a set of mathematical models which allow us to actually estimate these speciation and extinction rates. Is it a constant? Does it vary with the time? Does it vary with the diversity in that particular lineage which is present? Does it respond to the environmental variable? We use the paleo temperature as one of the predictor and use both canonical and pool diversification rates. What did we find? Indeed, angiosperms, as predicted in the literature, have much higher speciation rates as compared to the invertebrates and other vertebrates which were in our data set. Interestingly, where they come from and what are their biogeographic origins didn't have statistically significant difference. These are the Asian species or groups with Asian origins and a Gondwana origin. But what is really interesting is the variance associated with this, the Asian group. And again, habitat didn't seem to have an important role. But their evolutionary history, who they are, had a much more important role. When we fitted off these different models of constant growth, varying, and slowly saturating models, what we found, more than half of the lineages are slowly accumulating species. So they follow the gradual accumulation curve. The remaining 16 vary in their patterns. Some show time dependency, some show diversity dependency. So not all species across the groups are responding to these climatic changes similarly. Who they are, what traits do they have, seem to matter for the remaining half of these groups. Just to look at this same data sets in a different framework, we used factor analysis, and I'll go one by one. The first thing is, these were our 4 distinct scenarios. The gradual accumulation scenario is very distinct than the rest of the scenarios. Like species which are accumulating, or the groups which are accumulating species linearly, form a distinct cluster than the rest. You also have taxonomic groups segregating very neatly. Plants, herpetofauna and invertebrates form a very distinct cluster. So basically again, this showing who they are and their species traits really matter. Habitat types were overlapped. Asian and Gondwana lineages, they also had a kind of overlap, but if they are depending on the diversity within that group, had a very distinct clusters so as to they respond to the paleo temperature or not, also from 2 distinct clusters. And now that we understood how these groups have diversified in this landscape, we went back to our questions like, do this diversification rate really matter to the diversity gradient which we see now? People have again argued, if it's an older clade, you will have more species because more time for speciation. Versus if you have more opportunity to speciate, you will have higher diversification rate, and that would lead to higher species richness. For the western Ghats or the peninsular Indian group, what we see interestingly is that clade age, how old you are, really doesn't explain the species richness, but the net diversification rate is explaining the species richness patterns. To summarize this work, the historic stability of the landscape was very important. This landscape did face a significant geoclimatic events and it did change, it did travel, but there must have been the refugias within this landscape which acted as stable areas for diversity to accumulate. The remaining lineages 16 lineages did respond to payload temperature, Miocene acidification, ecological limits, the slowdowns which we see, and the species traits in the remaining groups. But it wasn't like we could identify which groups are responding to which. Like taxonomically, plants are doing this, or invertebrates are doing this, or arthropods are doing this. And the disparity in the diversification rate is a better predictor of what we see today as a species richness patterns. Where do we go from here? Today we need to think about how to incorporate fossils, because these are all the trees which are built using current existing species. They use fossils to calibrate the tree, but we don't have the fossils data while reconstructing these trees, and then we will get better estimates of speciation and extinction. We also argue that geography can play a very important role. How do these diversification rates vary with the geography and the climate? Is it interacting with each other? Now we see these diversification rates and the richness patterns are also telling us there are some bioregionalization patterns within this landscape, and what we don't know that yet. And also these processes which are occurring at a large scale, we are monitoring at a clade level or a group level. How do they be? The processes which are important at a micro level or a population level are the same processes will be important at macro level, or they are very distinct. And how does the speciation occur? Is it always adaptive or non-adaptive? These are the questions currently the lab is trying to address. And I would like to thank the entire lab, but also my mentors, Adam, collaborators here for this work. I'm happy to take questions, but I would like to take one more minute after that.
We have time for a couple of questions.
Really cool work, Jahnavi, the last part, the new work. I'm curious about this idea of potential refugia. I know it's theoretically predicted right now, but is there any way that you could make some predictions about that based on climatic models and so on?
Yeah, I think so, and there are some paleoecologists in this room, but we hoping to get some polar data, fossil polar data, and that can tell us which have been refugias, where are we seeing these species throughout the time scale, and then predict in geography where the refugias have been.
Then tested.
Yes.
Also, if I can ask another very quick question, wondering about, you said that there are some lineages which are not the gradual accumulating ones. Is there anything about those sets of lineages in terms of functional traits which might be different?
Yes, currently we are looking at that, but at the, on the surface it's not like herpetofaunal or invertebrate or plants. It's a mixed group, and now individual lineage traits is what we are going to look at to see if they have responded and how much they respond.
Yeah, that's a question.
Hello, very nice talk. So, as you said, if in this case it would be possible to maybe find out what the refuges were, and if we can understand during what climatic or geographic stress these refuges were used, can that be used as a prediction for future climatic stress?
Wonderful, and that's the hope here to identify which areas and what are the attributes of those areas which play an important role to act as a refuge through the geological history. I'll just take another minute because Karthik did say this is an event where we are going to think about how and what's the future of ecology. And I hope I was able to convince that the kind of biodiversity we have, and throughout this morning and in the images talk we have seen, what we are talking about is how evolutionary past has shaped the biodiversity. And Umesh very well illustrated that we are in the Anthropocene and global change drivers are changing the biodiversity which we see today. However, our understanding of the biodiversity as of now in India across its diverse geographies and ecosystems is very, very limited. It's not even 1 to 2%. And where do we go from now? So some of us have been thinking about, can we put collective effort to think about generating these data sets which are long-term which are using collections, distribution data, trade data, their evolutionary history, put together in one umbrella and think about how do we go and understand some of these challenges of biodiversity and actually contribute through data to talk about how the biodiversity is going to respond to some of the challenges it has. And I think this morning session, Rohit Nilekani asked, are we going to use climate change as an opportunity? Yes, but to do that, I think we really need to get a first very good handle on what is our biodiversity, where is it distributed, what are the basic tenets we can call about the biodiversity, which are how many species, where are they distributed, what are the distributional ranges, what traits they have. And I think that generating that baseline data for India across its ecosystems and geographies is extremely important. And I think this may be the starting point to think about a collective effort. And some of us have been thinking about it, how to go about it, how do you actually think and do it. Of course, you need to do field work. That's what is going to give you the data on the species and its distributions. You need to think about across ecosystems and not necessarily your favorite ecosystems and favorite taxa. You need to curate a huge number of collections to get to these data sets. And this will be a task not a one person or a one group or a one team is going to do. And we may need a more concrete efforts to do that. And some of us have been thinking about how to go about it. So we thought we'll use this forum to argue or emphasize the need for it.